and on behalf of my colleagues, Dr. Bill Wenling Zhang and Dr. Weizhong Wang. Bill is in Florida and Weizhong is in Ottawa. This is a biotech presentation about RNA, ribonucleic acid. I'm highly qualified to work in this field. I grew up in New York actually, and I went to Stuyvesant, then Cornell, and University of Washington, Seattle. I did my postdoc in Singapore, and I've been at Wayne State since '07, and this month I was promoted to a full professorship. I'm an NIH director as New Innovator. I got a New Innovator grant from Francis Collins a few years ago, and an internationally recognized expert and one of the pioneers in the area of long non-coding RNA biology. That's our team. Bill has genomics-driven drug delivery with reduced side effects. He's an expert on new classes of druggable targets. Weizhong is in Canada. He's really an expert with FDA filings, not only in North America, but also worldwide. The 20th century in biology was characterized by the advent of the central dogma. People thought that information only flows from DNA to RNA to protein. But in the 21st century, after the Human Genome Project was finished, people understood that protein coding genes are only 1.5% of the genome. So I participated in the ENCODE project, Encyclopedia of DNA Elements, and we counted human genes. We showed that non-protein coding RNA genes that are not translated into protein are more numerous, twice as numerous as protein coding genes. So these genes are key human disease regulators, but they remain very poorly understood. And that's our claim to fame. Most biotech pharma companies are still focused on conventional protein coding genes and on small molecule drugs to hit protein coding genes. Now, short interfering RNA therapeutics or siRNA therapeutics are a way to knock down the expression of any gene, coding or non-coding, and we always use those therapeutics in the lab. I cannot tell you everything in 10 minutes, but I will tell you a little bit about one of our targets. We have both a target discovery pipeline and a drug delivery pipeline. Target discovery pipeline, we already analyzed the whole human genome to find all long non-coding RNA genes that are capable of causing human disease. We validate those genes in the lab. In this particular example, we found one long non-coding RNA gene that's responsible for fasting glucose levels, which are a key diabetic trait. This gene controls another gene that converts blood glucose into liver glycogen. We have been able to target this gene with RNA-based therapeutics in the lab, and that's only one proof of principle. In vivo, in the body, you don't use siRNAs, you use modified backbone oligonucleotides, which are a very mature platform. So the innovative value of our project is twofold. First, we have targets. We already have a target that causes type 2 diabetes and obesity that's druggable, and also we have a pipeline to discover hundreds to thousands of similar targets in diabetes, cancer, and many other diseases. This could not have been discovered with our unique know-how, and this is really precision medicine. Instead of giving the same drug to everybody, we give sequence-based genomic drugs only to patients whose genomes contain the risk variant, risk allele as geneticists call it, in each long non-coding RNA locus. IP conditions are standard. We have patentable findings, and we do have one patent already being prosecuted now. Feasibility analysis, well, this technology is very feasible because we focus on the liver. The liver is the only organ in the body that can uptake modified backbone oligonucleotides injected into the bloodstream. This was shown many years ago by Santaris, which has since been absorbed into Roche. The novelty of our company is that we use genome-wide association studies and whole genome sequencing to discover personalized causes of disease that are specifically long non-coding RNAs and target them with sequence-based, personalized, rather than generic small molecule drugs. We have drugs against one target. What we now need to do is we need to scale this up to massively parallel deliver drugs against our multiple targets. It's very feasible. However, we need animal models, and we also need a scale up. And that is why we're here at the Nanning Biotech competition. I need to tell you that this is literally $100 million to $1 billion market. I'm not exaggerating. Think about type 2 diabetes, obesity, and cardiovascular diseases, especially in Western and more prosperous countries. These are non-communicable diseases of global importance. There is a huge unmet demand for better drugs, drugs that are personalized, drugs that don't have side effects that endanger compliance. We practice precision medicine, and the NIH in the US, as many of you know, currently really supports that approach. There are other companies in this field, for example, listed in the bottom of this slide, Alnylam, Ionis, and Regulus. All have siRNA-based drugs. None have our target discovery pipeline, and none have our target, which also is implicated in obesity and heart disease. Injectable siRNA-based or modified backbone oligotherapeutics actually work very well. I was here in Boston a few months ago at the SynGen Summit, happens every 6 months, highly recommended, and Greg Hickle from Alnylam spoke about their pipeline. In some of these cases, one lifetime injection is enough for a cure. And these are really cheap, easy to synthesize, easy to deliver drugs. And again, we're focusing on the liver. So our unique know-how converting genome-wide association studies to risk variants in long non-coding RNA genes that are druggable using rational drug design. That's what we bring to the field. RNA is a sequence-based drug. In a nutshell, these drugs, be they siRNA or modified backbone, are antisense to their specific target in the cell. Our targets in the cells are long non-coding RNAs, some of which have already been validated. These are informational drugs, not small molecule drugs, and their advantages are well known. So this project generates a pipeline of LNCRNA leads that are druggable. I know that cancer was also in the title. We will first focus on liver cancer for obvious delivery reasons, but the insights we're generating here are applicable not only to every cancer, but also to every human disease. So the social benefits of this project are quite obvious. As I said, we are proposing a totally novel way of handling chronic diseases that are already bankrupting first world economies and treating them with something other than antique, one-size-fits-all, imperfect drugs. In brief, we are seeking grant support followed by VC involvement with the idea of actually developing LNC March into a company that's suitable to be taken public. And since I do have a couple of minutes, I wanted to tell you a little bit about our technology. We have a unique computational pipeline that we developed in the ENCODE project that basically relates any genome-wide association studies, any whole genome sequencing, and in China, whole genome sequencing, PGI can do it now, $1,000 a person. We can collect this data in aggregate and figure out which non-coding RNA genes in the data are candidates, not just bystanders, not just near other genes. And then what we do is what the authors of all of those many papers you see in genetics journals don't do. What we do is we go to the lab and we work on real human cells, not crappy cancer cell lines, but primary cultures. The data I showed you was for primary hepatocytes. And we validate the function of our druggable LNCRNAs in these primary cells before we say that it's okay to advance from siRNAs to modified backbone and from in vitro to animal models. Here, we ask for your support in upscaling this technology so that we can move from siRNA to MBOs, from in vitro to animal models, and make personalized medicine a reality. Many of these variants are specific to one population. For example, African Americans, Europeans, Asian Americans have different risk variants. We are delivering population-aware therapeutics here. And lastly, Bill, Wei Zhong, and I would like to thank you all very much for your attention and for allowing us to present our RNA biotech pitch at the Nanjing competition. Thank you very much. We strongly encourage you to ask questions now and during the break. Thank you so much for having us.
As I understand, you have you said your portfolio is that your company, right? Your guys has organized a company that is a platform to have a portfolio of 4 products, right? And my question is, what's the relationship between these 4 products? They're complementary, they're alternative ways to cure something. That's one. Number 2 question is, what stage are these things?
Thanks, great questions. So, answer to question 1. The 4 products that that hit one target to cure one disease, which is type 2 diabetes, that's only an example of what we can accomplish. In fact, we have many other products against other targets that are being delivered. So that was merely proof of principle. What we really have, we have a target discovery pipeline that has already been implemented that's global, not only for diabetes, but for many cancers and many other diseases from genome-wide association studies. So those 4 siRNAs are simply one example. So the second question is, those are 4 sequences that work in vitro in primary human hepatocytes, liver cells in culture to effectively suppress the target that we need to suppress to normalize fasting glucose levels in the blood. So the next step with those particular sequences is to take them to animal models, mouse or rhesus, and then to human clinical trials. All the basic science in vitro has already been done.
Okay.
We have many more. We can upscale this to whatever extent funding is available.
So in the diabetes, in different stages, you have the weapons to target all this, right? Okay.
You're correct. We have weapons to target any long non-coding RNA that's directly causative in type 2 diabetes using this generalized target plus drug discovery pipeline and platform.
But what is stage, like clinical stages?
We are ready to go to clinical trials with one target, with the 4 drugs we have against the first target, but we need to develop other targets before we can do clinical trials for other targets. That's what we're trying to start and grow a company. So these 4, if we have funding for animal safety then clinical trials, we're ready now. Others, we need to develop. Thanks, great questions.
So, eventually, how long it takes to eventually a diabetes patient can take the drug? What do you, based on prediction, how long it will take?
You mean how long it will take us to develop, like from today to a drug?
Yeah.
Okay, so. So we, the 3 of us, discussed this question earlier today, and Bill's best case estimate is that it could be done in as little as 12 months. I know it sounds crazy and unrealistic, but please keep in mind, the drug platform, modified backbone oligonucleotides, already exists and has been used by many companies. It's our target discovery pipeline that's completely unique. So best case scenario, if you have accelerated human clinical trial, small sample size, we believe this could be ready for prime time for the target I showed you in as little as 1 year. For other targets, obviously it may need more time, maybe 18 months to 2 years, realistically. Thanks. And next question.
Now, you know, there's a Moderna here.
Yes,
so
I'm aware.
What is the major difference between the siRNA and the mRNA? Yeah, what is the major difference?
I know it. You don't need it. You don't need it.
So you don't need the...
Oh, I don't need the microphone.
Yeah, you don't need it.
Okay, sorry about that. Okay, so I don't claim to be comprehensively aware of every competitor's IP, but my impression is that Moderna started out as a company that does small RNA therapeutics, meaning like hitting microRNAs and endogenous small RNAs with RNA drugs. And I think that's great, that's very important. But small RNAs are a mature field. They get to be a little bit passé. It's a saturated field. The emerging field is long non-coding RNAs that are 10 times more numerous than microRNAs in the genome and in the body, and I would argue 10 times as poorly understood. So the difference between Moderna and anti-small RNA companies and us is that we have a similar approach. I mean, obviously the backbone chemistry is different, but the approach using a short antisense RNA is similar. We have a similar approach against a different and much bigger class of targets. And in fact, there is a lot of evidence to support the contention that longer RNAs are better targets, not only because they are longer, but because, interestingly, they are poorly conserved in evolution. So hitting them does not have as many side effects as hitting a microRNA, where when you suppress it, you may unintentionally alter other processes in the body that are not related to the treatment that you want to deliver. Okay. But they're a competitor, yes?
Yeah, thank you. But there took them a pretty long time, many years. And you think just 1 year you can prove your.
For the one target and the first 4 siRNAs, we believe, and I would be more than happy to sign an NDA with you to describe in detail how and why this is possible. We believe that it would take only 1 year from where we are today to human clinical trials well in progress of those first. No, we would need to do human clinical trials in partnership with one of Bill's collaborators. He runs a non-profit research center in Guangzhou, which I believe is licensed to do clinical trials. So potentially this would be something we would outsource to China. No, I would not be doing them at Wayne State. I'm a PhD, not an MD.
Okay. There's one more question is for the drug will be intake or injection?
Great question. So the bad news is that siRNAs are injectable drug. I'm not personally aware of oral siRNAs, although I heard rumors at conference people are working on it. But the good news is that in many cases, it turns out that one injection or a couple of injections is enough for many years worth of treatment or potentially a lifetime cure. And again, Greg Hinkle at Alnylam, which is by the way also based here in Cambridge, gave a talk at the Oxford NGS Summit here in Boston in May, and he said that some of their drugs, one injection delivers a lifetime cure, and the FDA has a problem with that because you need a clinical trial with a timeline series of injections to prove that you're ready for prime time. But if we're that good, why prove?
Yeah.
So yes, they're injectable, but they work well.
Yeah, injection is better, somehow you bypass the digestion system.
Yes.
But there's liver, do you have any, how the liver affect your drug?
So actually, injectable siRNAs, injectable modified backbone oligonucleotides, pardon me, I have corrected myself, injectable MPOs are very efficiently delivered to the liver. They do not suffer there from interferon response, RNases, or side effects. And in fact, the very first proof of principle, Santaris, a company in Denmark that was subsequently acquired by Roche, they delivered by injection, straight to the injection and in the bloodstream, and it was delivered to the liver, oligos against the microRNA target, speaking of Moderna, MER122. And MER122 is a hepatitis C virus cofactor, so they stopped hepatitis C virus replication by delivering, by injecting into the blood these MBOs that went to the liver. So I appreciate your concern, but many companies have proven that the contemporary chemistries that I showed on my slide, History of Oligotherapeutics, are capable of going around these problems. So yes, it's possible. And lastly, profit margin. Oligos are so cheap to synthesize. I have a friend in Sydney who synthesized them on his garage synthesizer. It costs $100, but you can deliver it as a $100,000 drug if it's a lifetime cure for some disease that's in demand. That's the profit potential here. That's why I say it's a $1 billion market here.
Yeah, I believe that the diabetes is a very big disease. If you can successfully cure that, diabetes, that's great contribution.
We believe sincerely that we can cure it in patients whose diabetes risk is primarily underlined by this locus. Of course, not in patients whose risk variants are at other loci, but that's the meaning of personalized medicine. So to that extent, yes, we believe it.
Yeah, how much you are seeking investment?
Yeah, so I'm sorry, actually the version of the presentation here was not the latest, but that's absolutely okay. You did a fantastic job, it's okay. So I sent a couple of slides earlier that actually didn't show up. We believe that we would need approximately, we would need 1 to 1.5 million US dollars to get started. So preferably 1.5 million USD, so that's like 10 million RMB. And that's an R01's worth of money. So I guess that's a lot if you're an academic. It may not be a lot if you're in industry. That's what it would take us to deliver the 12 months with the first 4 oligos and 1 target and get the other leads, of which I have many. Just sign an NDA, we'll show them all, and take our other leads to in vitro validation. So 1.5 million would be good, yeah. 1 to 2 years.
So what kind of hypothesized model you used, and what is the control? And how long you be monitored efficacies in the cell groups?
Okay, so in our primary hepatocyte cell cultures, there is a simple time limit because, of course, human hepatocytes do not divide in culture. They live for 5 days and then they die.
No, that's not true. They can live for 2 months if you have a good monitor.
okay, if they can live for longer than 5 days, that's true. But in my technician's hands in our lab, we've only been able to do 5-day timelines. I'm sorry, and thank you for the correction. So in this case, everything was done inside 120 hours. So the manuscript, I want to say manuscript isn't correct. The manuscript has been submitted to one of the major diabetes journals, everything was done in biological triplicates, of course. So we have 4 different siRNAs against one LNCRNA target, the LNCRNA target we found from GWAS. So each of the 4 siRNAs was done in biological triplicates. There were also scrambled siRNA controls and mock transfection controls that did not involve an oligo. So those 2 kinds of negative controls were also done in biological triplicates. Also, because this is a population-specific disease and it's not trans-ethnic, in this case, we did experience on both European American origin and African American origin hepatocytes, because in African Americans this is not a risk locus, and in Europeans it is. So in brief, those were the numbers of triplicates and controls included in the cell culture work.
So you only use the normal livers, not disease liver?
Yeah, we're greatly limited by commercial access to hepatocytes, which we have to purchase from Lonza. So it is not true that all of those are normal livers, but I would say it is true that either those livers came from patients who had type 2 diabetes, or they came from patients who did not have it, but whom we call normal, because, for example, they died of other causes. They may have had alcoholism, cirrhosis of the liver. I mean, obviously it's not good, but they were not diabetic. If we can succeed in getting funding for this project, the first thing we will do is we'll actually properly compare diabetics and controls without being limited by Lonza in terms of the kinds of cells and the donors that we can find. So that's a limitation of the project, I admit that. That's preliminary data that was generated using my NIH New Innovator report. Thank you for your question, for your critique. I appreciate it.
Okay, thank you so much.
Thank you for hearing. I appreciate it.
Really well delivered and very informative.
Thank you, sweet.
Thank you. So, okay, thank you so much to judges. 我们现在就进入到这个午休的时间，我们有1个小时，然后在1点半的时候回来，我们会进行下午场的这个项目调研。
对，然后我们这个餐马上就到，应该是。
已经到了，已经到了。
好，那我们现在就可以出去吃了。
我们为大家准备了午餐，然后。